Biochemistry 401 Lecture 22. Today we're going to talk about ATP synthesis. We're then going to talk about the uncouplers of oxidative phosphorylation and the poisons that can interrupt the flow of electrons in the electron transport chain and ATP synthesis. So let's get started. The energy that's released during electron transfer can be used to pump protons into the intermembrane space. This creates a proton concentration gradient and also electrical potential across the inner membrane. This is called a chemiosmotic gradient or chemiosmotic potential. Chemiosmotic potential causes protons to flow back into the mitochondrial matrix, which turns a turbine-like molecular motor, which helps to synthesize ATP. And so what we're doing is we're pumping protons from the matrix into the intermembrane space. And we have a separation of charge here. We're loading the inner membrane space with chemiosmotic potential. And this is similar to a battery. In this diagram, we see three different representations of ATP synthase. In all cases, the intermembrane space is at the top of the slide, and the matrix is at the bottom of the slide. On the extreme left, we see a cartoon diagram of ATP synthase. And we can see that it is comprised of two main regions, F0 and F1. The F0 region is a place into which protons will flow, whereas the F1 region is a portion of the enzyme that actually synthesizes ATP. Now, this enzyme uses proton flow to cause a portion of this enzyme to actually rotate, and this rotational energy helps ATP synthesis. Let's look for a minute at the diagram in the center, which is from your book. This shows that F0 and F1 are actually comprised of many subunits. And so there are two different types of components in ATP synthase, those that rotate and those that don't. The rotating components are the C ring and the gamma and epsilon region of the stalk, whereas the stationary components are the stator. This is comprised of A, beta 2, and delta subunits, and the alpha 3, beta 3 hexameric ring also is stationary, and this is because the stator is keeping it still. The stator is anchored in the inner mitochondrial membrane by the A region, and the B2 region forms a link between the A region and the delta region. It is the delta region that makes physical contact with the alpha 3 beta 3 hexameric ring and keeps it still. Now the C ring rotates within the plane of the membrane, and as it does, it causes the drive shaft, the delta region, to rotate as well. And it is protons that are moving from the intermembrane space to the matrix that actually cause the C ring to rotate. The protons do not go down through the central channel as one would expect, but rather the protons are transported across the inner mitochondrial membrane by individual subunits of the C ring, and we'll see that in a minute. And so let's go over this really quickly. What's going to happen is this. Protons are going to cause the C ring to rotate. The rotation of the C ring is going to cause the central shaft to rotate as well. This rotation is going to cause a conformational change in the beta subunits of the alpha 3 beta 3 hexameric ring. And this conformational change is going to open and close regions in the beta subunits, allowing ADP and inorganic phosphate to enter, to form ATP, and for ATP to exit. Now let's look at this a little more closely. Again, here we see the three-dimensional structure of ATP synthase. Now, if you'll notice, the whole of the stator is not included. That's because it's a very difficult portion of the protein to actually crystallize. and so all we see is the A region of the stator. Now the gamma subunit acts as a central shaft, and it rotates within the hexameric ring. But you can see that this central shaft is not symmetric, and it is this asymmetry that causes conformational changes in the beta subunits. Here we see the central shaft as it lies in the middle of the hexameric ring. Portions of this ring have been stripped away so that you can see the gamma subunit in the center. As this gamma subunit rotates within the hexameric ring, it causes conformational changes to occur in the beta subunits. And here again, we see how the alpha 3 beta 3 hexameric ring surrounds the gamma subunit that comprises the central shaft. And if we rotate the hexameric ring 90 degrees to look at a cross-section of it, we can see the central channel into which the gamma subunit extends. And so we've said that rotation of the C ring causes the gamma subunit to rotate as well, and this causes a conformational change to occur in the beta subunits that help to power ATP synthesis. But what causes the C ring to rotate, and how do the protons go across the inner mitochondrial membrane? Well, this happens through the action of the subunit A. This contains two half channels, one that has direct contact with the cytoplasm and one that has direct contact with the matrix. However, neither of these channels spans the membrane, but instead will abut a C unit. This means that it has direct contact with the subunits of the C ring. Now each C subunit contains a critical aspartic acid that is important for C ring rotation. Now we know that aspartic acid can act like an acid to lose a proton, and this residue has a key role in delivering protons from the inner membrane space to the matrix and also allowing the C ring to rotate. Let's see how that works. And so remember, the intermembrane space has a high concentration of protons. Therefore, the pH is low compared to that in the matrix. In the matrix, there is a low concentration of protons, and so the protons will move down their concentration gradient, and this powers ATP synthase. Here we see a cross-sectional representation of the C ring, in which the protein backbones of each subunit is shown. We can get a good appreciation for the individual subunits this way, and we can see quite easily the aspartate residue that's indicated by the arrow. This aspartate residue is going to pick up a proton from the intermembrane space. Protons will flow in to the cytoplasmic half channel in subunit A, and once there, they interact with the aspartate residue in the C subunit. Now, aspartate in a proton-poor environment will be deprotonated to form aspartate. This is a negatively charged amino acid. However, in a proton-rich environment, at a pH much below its pKa, this residue will be protonated to form aspartic acid. Now, aspartic acid is neutral, and so it can enter a hydrophobic membrane quite easily, whereas the aspartate cannot. The presence of aspartate in the C unit, rather than aspartic acid, will prevent the ring's rotation. However, once aspartate is protonated to form aspartic acid, the C subunit can freely rotate. And so here we see a schematic that represents the A subunit as it interacts with the C ring. We can see the half channels illustrated quite nicely in the front, whereas the C ring is shown behind it. Protons from the intermembrane space are going to flow into the cytoplasmic half channel down their concentration gradient. Once there, They will interact with aspartate to form aspartic acid. And the C ring will rotate all the way around until that particular subunit reaches the matrix half channel. Now the matrix, you'll remember, has a lower concentration of protons than the intermembrane space. And so aspartic acid will become deprotonated again. The protons are released. The beta subunit is the site of ATP synthesis, and it has three conformations: loose, tense, and open. In the loose state, ADP and inorganic phosphate bind. This is shown in the blue section in the diagram to the right. In the tense conformation, ATP is synthesized, and in the open conformation, ATP is released. Now, it was shown that the rotational energy from the gamma subunit is not necessary actually for ATP synthesis to occur. ATP synthesis can occur without the proton motive force that causes the gamma subunit to rotate. What cannot happen, though, is for ATP to exit the beta subunit. The open conformation relies on the conformational change that happens when the gamma subunit rotates. So let's go through this bit by bit. The subunit that is highlighted in yellow is in the tense conformation because of its interaction with the central gamma subunit. In this conformation, ATP will be synthesized. As the gamma subunit rotates, it's going to cause the yellow beta subunit to now be in the open conformation. ATP is released, and another ADP and inorganic phosphate combined. The electron transport chain, under normal circumstances, is coupled to ATP synthesis. The passing of electrons down the electron transport chain provides the energy to establish a proton gradient, to provide the proton motive force for ATP synthesis. But there are things that are called uncouplers that provide another way from the intermembrane space to the matrix, circumventing ATP synthase. One such molecule is called UCP1. This is also called thermogenin. This is a protein that provides direct access of protons from the intermembrane space to the matrix. Now, instead of all this energy that's being released by releasing the proton motive force, being used to make ATP, this energy is used to make heat, and this is reflected in UCP1's other name, thermogenin. Thermo is heat, and genin is to make. Thermogenin is an important component of brown fat. This is fat that is especially prominent in mammals that hibernate and in human infants, and the role of this brown fat is to help regulate body temperature. And so when the body is responding to cold, fatty acids will be broken down, and this will upregulate the activity of UCP1 and allow protons to flow from the intermembrane space to the matrix. There are other uncouplers as well, and one of these is 2,4-dinitrophenol. This is a small compound used to make TNT. Persons who were making TNT in dynamite factories would often become poisoned with this, as 2,4-dinitrophenol takes protons directly from the intermembrane space to the mitochondrial matrix, thus bypassing ATP synthase. You can imagine the symptoms for poisoning by this molecule: high temperature and lack of energy. There are other poisons that can interfere with ATP synthesis. Some of these have effects on the electron transport chain, and others have an effect on ATP synthase directly. It is important for you to know these. You will be expected to know them. Complex 1 is blocked by rotenone and amytal. Complex 3 is blocked by antimycin. Complex 4, cytochrome oxidase, is blocked by cyanide, azide, carbon monoxide, and H2S, hydrogen sulfide. ATP synthase itself is blocked by oligomycin. Rotenone is an insecticide and amytal is a barbiturate. Both of these can block complex 1. Complex 3 can be blocked by antimycin, which is an antibiotic. And again, complex 4 is blocked by cyanide, azide, hydrogen sulfide, sewer gas, and carbon monoxide. ATP synthase is blocked by oligomycin, which is an antibiotic. And so there are a lot of poisons that poison the electron transport chain and ATP synthesis. Pretty nasty stuff. Now, if you were to block the electron transport chain by any of the poisons that block complex 1, 3, or 4, you would eventually stop using oxygen because oxygen would no longer be needed as the ultimate electron acceptor. The electrons will be blocked at a point before they even get to oxygen, and so oxygen consumption by the mitochondrion will go down. If you were to block ATP synthase by using oligomycin, oxygen consumption would remain the same for a while until the concentration of protons would be so great that the complexes would no longer be able to move protons against the concentration gradient because the concentration of protons in the intermembrane space will be too high. This can be relieved through the use of an uncoupler that will relieve the chemiosmotic pressure by allowing protons to flow from the intermembrane space into the mitochondrial matrix. However, ATP will not be produced. Electrons will be passed, oxygen will be consumed, but ATP will not be produced. So how much energy do you get from NADH and FADH2? This can be measured in what's called a PO ratio. This stands for the ratio between moles of ATP produced per moles of molecular oxygen reduced to yield water. NADH produces 2.5 ATPs per oxygen, and FADH2 produces about 1.5. So for every NADH that enters the electron transport chain, you get a resultant 2.5 moles of ATP, whereas for FADH you only get 1.5 moles. But why is it that you only get 1.5 moles of ATP per mole of oxygen used if you're starting at FADH2, whereas you get 2.5 from NADH? Well, if we look at the electron transport chain, we can see that for every NADH that comes in, more protons are actually pumped into the intermembrane space. If you're coming in at the position of FADH2, you're losing the proton motive force that would have come in from complex 1. And so, since there are less total protons that are being moved from the matrix to the intermembrane space, when you're starting with FADH2 than if you would start with NADH, less moles of ATP are synthesized per mole of oxygen used. So how much ATP do you get from the complete oxidation of glucose? In glycolysis, we're going to convert glucose into pyruvate in the cytoplasm. You lose one for the phosphorylation of glucose. You lose another one for the phosphorylation of fructose 6-phosphate. But you gain 2 with the dephosphorylation of 2 molecules of 1,3-bisphosphoglycerate, and another 2 for the dephosphorylation of 2 molecules of phosphoenolpyruvate. This leaves you with 2 net ATPs, and you also get 2 molecules of NADH from the oxidation of 2 molecules of glyceraldehyde 3-phosphate. Now, from the conversion of pyruvate into acetyl-CoA inside the mitochondrion, you get 2 molecules of NADH. And in the citric acid cycle inside the mitochondrion, you get 2 molecules of guanosine triphosphate and this can be thought of as ATP. You also get 6 molecules of NADH and 2 molecules of FADH2. And so we get 2 net molecules of ATP from glycolysis from each mole of glucose, and 2 ATPs from the TCA cycle per mole of glucose. This yields 4. But we also have reducing equivalents. We have 2 molecules of NADH formed in glycolysis, and if we use the glycerol 3-phosphate shuttle, each will yield 1.5 mole of ATP. If, of course, we use the malate aspartate shuttle, that will be 2.5 moles of ATP per NADH, because we won't have to go down to FADH2. We get 2 molecules of NADH formed in the oxidative decarboxylation of pyruvate, and each will yield 2.5 molecules of ATP. 2 molecules of FADH2 in the citric acid cycle, and so we'll get 1.5 molecules of ATP from each of those, and 6 molecules of NADH in the citric acid cycle, and we'll get 2.5 molecules of ATP from those. And so, if we use the glycerol 3-phosphate shuttle, we'll gain 30 molecules of ATP per molecule of glucose. If we use the malate aspartate shuttle, we'll have 32 molecules of ATP per molecule of glucose. And so this marks the end of Lecture 22. Have a super day.